I'm going to report an article called Inferring the Genetic Variability in Indian SARS-CoV-2 Genomes Using Consensus of Multiple Sequence Alignment Techniques from the journal Infection, Genetics, and Evolution. Severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2 causes a disease COVID-19 which originated in Wuhan, China. As we all know, this disease has brought a wave of pandemic throughout the world, and vaccine is the primary prevention strategy to contain this virus. However, the development process of vaccine is time-consuming and requires the analysis of genetic variability of the virus population so that effective and safe vaccine can be developed. This 2020, Tunghan has performed a genomic analysis to show the evolution of SARS-CoV-2. Saha et al. Also analyzed Indian SARS-CoV-2 genomes. To continue their works, the author of this article performed genomic analysis of 566 SARS-CoV-2 virus population to identify the mutation as substitution, deletion, insertion, and single nucleotide polymorphism or SNP. SNP is the 1% of the population being affected by substitution. Only non-synonymous mutations are responsible for the changes in amino acids. To find the genetic variability, multiple sequence alignment or MSA is needed in presence of reference sequence. This technique provides a near optimal result, thus these different alignment techniques can provide different results on the same pool of sequences. So the researchers used four different well-known multiple sequence alignment techniques, which are Clustal W, Muscle, Clustal O, and MAFT. They used these in order to align the Indian 566 SARS-CoV-2 sequences, and those aligned results are used to identify the lists of mutations as substitution, deletion, insertion, and SNP. So as said on the previous slide, these assigned results are used to identify the list of mutations as substitution, deletion, insertion, and SNP. Consensus Multiple Sequence Alignment or CMSA is prepared to have the final list of mutations. This is to preserve the advantages of all four alignment techniques. Identification of SNPs can be helpful to classify virus strain, design vaccine, and identify the dose of the vaccine. The metagenomic analysis using next-generation sequencing or NGS shows that the SARS-CoV-2 is a single-stranded envelope RNA virus with a genome length ranges from 27 to 32 kilobases. As reported in NCBI, SARS-CoV-2 has 11 coding regions that can encode ORF1 lab polyproteins, spike S glycoprotein, envelope E protein, membrane M glycoprotein, nucleocapsid N protein, and 6 more accessory proteins. This ORF or opening reading frame can encode several non-structured proteins or SNP. So as shown in this slide, this is the overall pipeline of the research or the genomic orientation of the SARS-CoV-2 virus. So to input the SARS-CoV-2 genomes, they collected the recent genomic sequences of Indian SARS-CoV-2 virus from the Global Initiative on Sharing All Influenza Data or GIS-8 in FASTA format, which contains 566 complete and near-complete genomes. The average length of the 566 genomes is 29,831 base pairs. Next, they downloaded the reference sequence from NCBI and used BioEdit for the data visualization and editing. So to prepare the consensus genome from aligned set of genomes, they applied four different multiple sequence alignment techniques, such as Clustal W, Muscle, Clustal O, and MAP, to align the 566 SARS-CoV-2 sequences. Clustal W, Clustal O, and MAFT are progressive alignment techniques, while Muscle is an iterative progressive alignment technique. Progressive alignment technique use Needleman-Wunsch algorithm, Smith-Waterman algorithm, and so on to complete the pairwise alignments, and then the sequences are clustered together to show their relationships by using the methods like k-means algorithm. The iterative progressive alignment technique works like progressive alignment technique, but dynamic programming is repeatedly applied to realign the initial sequences. So here shows the preparation of consensus genome from the aligned set of genomes per method. Clustal W initially performs pairwise alignment of all the sequence by using k-tuple method. Then MSA is created by progressively aligning the most closely related sequence based on the neighbor joining guide tree method. Clustal O uses the k-tuple method to produce pairwise alignment, then embed is used to cluster the sequences, followed by k-means clustering algorithm. Then the guide tree is built using the unweighted pair group method with arithmetic mean or UpGMA method. Lastly, MSA is constructed by using the HH-align package. Math uses two different methods, the progressive and iterative refinement. The objective of math is to merge local and global algorithms for MSA. Initially, progressive refinement is used to calculate all pairwise distances to create a provisional MSA from which refined distances are calculated. Then iterative refinement is performed to get the final MSA. In muscle technique, they use two distances measure, which are the k-mer for aligned pairs and Kimura method for aligned pairs of sequences. Then a draft MSA is produced in using muscle, which uses the k-mer method. So after that, a progressive alignment is constructed based on the guide tree as produced by the UpGMA method. This initial tree is re-estimated using the Kimura distance method. Then UpGMA method is used to produce a new guide tree which creates a second MSA. The new MSA are finally created by realigning the two sequences created previously. These four techniques were used to align, and using these aligned sequences, they identify the mutation in Indian virus sequences. These methods and the subsequent identification technique provide lists of mutation as substitution, deletion, insertion, and SNPs. Consensus results called Consensus Multiple Sequence Alignment or CMSA is prepared to have the final list of mutations so that the advantages of all four alignment techniques can be preserved. This table shows the mutation results of different methods on Indian SARS-CoV-2 genomes. CMSA identifies unique 767, 2025, and 54 substitutions, deletions, and SNPs, while the zero insertions are common among the four methods. All mutations from CMSA amount to 2,792, which came from its substitution, deletion, and insertion combined. Here shows the results of the methods and CMSA, and this is the detailed results for Table 1, which shows the mutation of SNP present in more than 10% of the SARS-CoV-2 genomes. It contains coordinate positions of the mutation, number of occurrence of frequency of mutation in the virus genome, type of mutation, change in the nucleotide and amino acid, entropy to measure the change, and genomic location and mapping with the coding. So all of these figures are examples of mutations like substitution, deletion, and SNP. This only shows the Venn diagram for the four MSA techniques or visual presentation of the tables. This figure shows the few examples of substitution, SNP on the top, and on the bottom shows the deletion. They use bioedit software to make this. Here shows the results of CMSA and are also visualized by using biocircus plot. Whole virus genome with the coding regions is shown in the outer track while substitution, deletion, insertion, and SNP are visualized in subsequent tracks where frequency of mutations is illustrated to the bar, which are the light yellow, orange, and violet, and the dot plots, which is the black dots. This gives a complete visual representation of the frequency of mutations. For example, in case of SNP at the coordinate positions of 241, 3037, 14,410, and 23,405, the change in nucleic acid is close to 60% of the Indian virus population. Out of these four major changes, two of them are belonging in ORF1 lap and one in the spike gene. Apart from that, ORF3A, membrane, and ORF8 nucleocapsid genes are also having SNPs. This chart represents the SNPs present in more than 10% of the Indian SARS-CoV-2 population at SNP coordinates position of 241, 3037, 14,410, 23,405. The change in nucleic acid is close to 59 to 60% of the Indian virus population. So this study shows the genetic variability of Indian SARS-CoV-2 genomes using consensus of multiple sequence alignment techniques. The analysis shows 767, 2025, and 54 unique substitutions, deletions, and SNPs are present in Indian SARS-CoV-2 genomes. More precisely, out of 54 SNPs, 4 SNPs are present close to the 60% of the virus population. These mutations are non-synonymous in nature, so this frequent mutation needs to be studied in the future. Finding SNPs is mainly to recognize the genomic locations that can be used to categorize the virus strain in India. So once the strain of the virus is identified, the proper vaccine can be used, and these SNPs can be used in the future for protein modeling so that drugs can be designed to target such proteins. The title of the research article made an impact on its first read, which makes it a good title. It offers a brief cover on what to expect when the article itself is read. It is not complicated to interpret and stimulates the reader's interest, especially today, which makes people curious about COVID-19. The abstract is of perfect length, which makes it a perfect summary for the whole research article. The objective of the paper is clearly defined and direct to the point. The qualitative data and tables are well presented, as well as the diagrams, which are visually appealing. It is great that it has sections for ethics approval, consent to participate, availability of data, supplementary materials, funding, and acknowledgement. Major findings were addressed and the conclusions were logically stated. The downside of this paper is that the biocircus plot, which was presented on the result as a diagram, should have been included on the methodology part. The researchers did not include how they operated the software as well as the four multiple sequence alignment techniques. Overall, this research article is interesting and has a significant contribution in the field of finding more studies about the recent COVID-19.